I'll use this one. That was interesting. So we'll just get going in 1 minute. Good. Well, good morning, everybody. Welcome to Functional Metagenomics 2022. My name is Trevor Charles from the University of Waterloo and Waterloo Centre for Microbial Research. We are coming to you today from the Pillar and Post Hotel and Conference Centre in lovely Niagara-on-the-Lake, Ontario, Canada. Today's program is a hybrid event with both in-person and virtual presenters and attendees. First, I would like to acknowledge the support and sponsorship of the Research Council of Norway through the Norris Casa project, the EU Microbiome Support, and the Waterloo Centre for Microbial Research. We are grateful for this support. This series started over 10 years ago, and there have been events in Canada, South Africa, and Norway. In this return to Canada, this current event was postponed from 2021 for obvious reasons, but we are thrilled to be back in person. I'd like to start with a land acknowledgement. I acknowledge the land on which we gather today is the traditional territory of the Haudenosaunee and Anishinaabe peoples, many of whom continue to live and work here today. This territory is covered by the Upper Canada Treaties and is within the land protected by the Dish with One Spoon Wampum Agreement. My workplace, the University of Waterloo, is on the traditional territory of the neutral Anishinaabe and Haudenosaunee peoples. Our main campus is situated on the Haldeman Tract, the land promised to the 6 nations that include 6 miles on each side of the Grand River. Furthermore, I acknowledge the lands on Treaty 1 territory of the Anishinaabe, Cree, Dakota, Dene, and Oji-Cree peoples that sustained my settler and indigenous ancestors and are the heartland of the Métis people. I acknowledge the lands of my birthplace, the unceded territories of the Musqueam, Squamish, and Tsleil-Waututh nations. And finally, I acknowledge the lands of the Kalinago and Taino, to which my African ancestors were brought. It is important to recognize the history of the lands that we inhabit. So today, I have the honor and privilege to introduce Professor Joe Handelsman as we kick off Functional Metagenomics 2022. Professor Handelsman did her undergraduate training in agronomy at Cornell University and her PhD in molecular biology from the University of Wisconsin-Madison. She spent much of her career in the Department of Plant Pathology in Madison, then spent some time at Yale University. From 2014 to 2017, she was Associate Director for Science in the White House Office of Science and Technology Policy, and has since returned to Madison as Director of the Wisconsin Institute of Discovery, a transdisciplinary research institute exploring information across disciplines at the interfaces of research, education, and business. Throughout her career, in addition to running a dynamic and influential research program in the areas of plant microbe interactions, metagenomics, which is a term that she coined, and the microbiome in agriculture, environment, and human health, Professor Handelsman has been a tireless advocate and leader in science education, mentorship for women in science, and for science policy and the role of science in society. As scientists, we need to never lose sight of the wonder of science and the role that it plays in making our world a better place. Please join me in welcoming Professor Handelsman to Functional Metagenomics 2022.
Well, hi, everybody. It's great to be here virtually. I tried very hard yesterday to get to you, but unfortunately, Delta Airlines wasn't cooperating, so I couldn't quite get there. So I came back home, and there were very few compensations for spending 5 hours in the airport and then finding out that I wasn't going to be in beautiful Canada. Except there was one thing that happened, and I wanted to share it with you that I thought was pretty cool. Let's see if you can see this. And I apologize for the video quality is not great on screen for some reason. Can you see the deer? Yes? No? Yeah? Okay. So yesterday, a little fawn was born outside my window, and this is it hanging out with its mom. And I sort of spent the rest of the day, instead of traveling, watching the fawn and its mother get to know each other. So that was my compensation for not being there, which is minimal, but it was something. So anyway, I hope you enjoyed my fawn. Okay, well, now I would like to turn to my talk, and I'm really excited to be here because functional metagenomics was really not an enormously popular word 20 years ago. And so it's really exciting to see that people are recognizing the power of the functionality of metagenomics and not just the power of sequencing. And so I thought I would tell you a little bit about the history of metagenomics and when and why some of the ideas that ultimately became my version of functional metagenomics came to me. So in the 1990s, my lab was working on plant pathology problems, mostly looking at suppression of disease by biocontrol agents, bacillus strains that live in the soil and on roots and can suppress diseases of plants caused by oomycetes. And that work led to the discovery of an amazing antibiotic, zwittermycin, which is a hybrid polyketide NRPS, or non-ribosomal peptide. And it has tremendously diverse activities, from augmenting the activity of Bt toxin against insects to many different antibiotic activities. And its synthesis actually follows a pretty dramatically new pathway. So we were entrenched in discovering zwittermycin and other antibiotics. We were also performing our first 16S-based analyses of the uncultured communities in soil and being overwhelmed, as everybody was and probably still is, by the tremendous diversity of uncultured organisms in soil. At that time, we were estimating that less than 1% of the organisms in soil were readily cultured, and we were finding clades of organisms from archaea to acidobacteria that had not yet been cultured. And so it was a pretty exciting time in biology generally, because Norm Pace and others had just opened up the world of the uncultured. But it was also a very difficult time for me, because it was a time when my mother was dying over a 16-year period of persistent lung infections. They were recurring lung infections. We never quite found out the basis for it. She seemed to have some kind of unexplained immune suppression that made her vulnerable to these infections. And she would be well for a while, then she would get a Pseudomonas infection or a Burkholderia infection. They would treat her with antibiotics. She would be better for a while, and then the same antibiotic wouldn't work because of antibiotic resistance. And so it was kind of a torturous time for me, because here I was discovering antibiotics, but my mother was running out of antibiotics, because one by one, they fell off the list of options because of resistance. And so I was sitting and talking to a colleague, Bob Goodman, here at the University of Wisconsin one day, and I was just sort of wringing my hands, saying that all of those diverse organisms down in the soil must have tons of new antibiotics. There's no reason to imagine that the diversity of antibiotics is somehow sequestered to the cultured organisms, which of course is a completely human construct. And so in the course of this conversation, we suddenly thought, well, perhaps we could take the pathways out of these bacteria without culturing and clone them into surrogate hosts, and maybe we could get expression of these new antibiotics. And so that became called functional metagenomics. And we never actually found too many antibiotics by that means. It turns out to be a far more difficult proposition than we thought it would be. But my lab went on to study antibiotic resistance, and particularly in the environment. And that's what I'd like to talk to you about today. But I think it's important to remember the origins and why these ideas come up, and how sometimes the tragedies of life can also drive the discoveries and creativity in science. So I'm sure it's not lost on most of you that we have pretty high level antibiotic exposure in many environments. In animals, their antibiotics are used for growth promotion, prevention of disease, and of course, treatment of disease in many livestock habitats. In plants, they are in a few cases used to prevent disease. The big one in Wisconsin is prevention or treatment of fire blight disease, which is a bacterial disease of apple trees. And you can see the plume of spray here. That's actually streptomycin being sprayed into the atmosphere. And this is particularly of interest to me because streptomycin is one of the antibiotics used to treat human tuberculosis. And a strain of Mycobacterium, not TB, but a related one, was actually one that infected my mother, and she was on streptomycin for a period of time. And so the idea of spraying vast amounts of antibiotic into the atmosphere and selecting for resistant organisms is really quite atrocious to me. So we began to study antibiotic resistance by metagenomics in two different kinds of environments. Some were where there's high level exposure near the target of the antibiotic. So in this case, in Connecticut, when I was at Yale, we studied a cow operation, dairy operation, where cows were repeatedly treated for mastitis with antibiotics. We also studied apple trees in Wisconsin before that, but I'm not going to talk about those results. I'll just talk right today about the manure. And then we also studied areas where there's presumably low level exposure to antibiotics in soil. And we assume that that's the case because antibiotic producing organisms are abundant in soil. That's where most of our antibiotics in clinical use come from. And so presumably some of them are producing antibiotics at low levels. We also imagine that most environments on Earth have some sort of low level exposure to antibiotics just because of movement of antibiotics throughout the environment. So I'll talk about several environments that we studied. One is in Alaska, a site where we're studying the bacteria in the soil on this island in the Tanana River. So it's pretty isolated from human activity. Besides a few swimming animals, it's pretty sequestered to birds, microbes, and organisms that reproduce on the islands. So there's very little human contact other than the researchers that go there, including my group. And so that seems like a pretty low level exposure environment. This is a medium level to high level. This is the West Madison site. I showed you the cow site, the dairy operation, which is the high level. And then this is a saltern in Puerto Rico, and I'll talk about that as well. So I'll start with the effect of manure on antibiotic resistance, and then talk about two other somewhat surprising discoveries we made. So I don't think this group needs to hear the process for doing functional metagenomics. We simply used a very powerful extraction of DNA from the soil that was also gentle enough to generate very large fragments of DNA. And for some of this work, we generated back inserts, back libraries with inserts of 60 to 80 KB. And then for much of it, we used phosmids and other vectors that would accept much smaller inserts. And all of the work I'll talk about today was in the host, the surrogate host, E. coli. And so after creating these libraries in E. coli, we would then screen the transformants by selecting on various antibiotics. So if we start with the manure treatment, these are cows that typically get mastitis at some point in their milk producing lives. And during that time, they cannot be milked because they are contaminated with bacteria, but of course they're also contaminated with antibiotics. And the most common treatment, at least in Connecticut at that time, was to treat cows with various beta-lactam antibiotics. And so these cows were mainly treated with cephalosporin type beta-lactams. And then we looked for antibiotic resistance genes in the manure from the cows that had been treated, which we thought would be the highest source of antibiotic resistance. And then we also followed the manure into the soil in a field site and looked at the impact of antibiotic resistance genes in the soil that was fertilized with the manure. So when we looked at the treatment of the soil in particular, we did find some antibiotic resistance genes, resistance to cephalothin, which is the antibiotic of choice for these cows. There was resistance in the manure, but not vastly high levels. But what we found in the soil was a really interesting phenomenon. And if you focus here on the right panel, we're looking here at the CFUs of resistant clones that are resistant to cephalothin. And we found, I'm sorry, this is bacterial populations here, not clones, not metagenomic clones. And what you see is the gray represent where the soil has been treated with manure. The black is soil that's just been treated with a chemical fertilizer, not manure at all. And what you can see is that across those treatments, there does seem to be across for 130 days, at most samplings up to about 94 days, we did find higher levels of antibiotic resistant, cephalothin resistant bacteria in the soil that had been treated with the manure. So we thought that for sure there would be higher levels of cephalothin resistance because the manure had been added. Either there was still antibiotic in the manure, or from the treated cows, or perhaps there were resistant organisms that had been selected by the antibiotic in the manure. But one suspicious thing that we found was on the left-hand side, you see total CFUs. And you can also see that total bacterial counts in the soil from the manure-treated soil, so those are the gray bars, were also significantly higher. So that suggested that under manure treatment, there were simply more bacteria, not just more beta-lactam resistant ones. So that got us wondering what was going on here and what was the impact of the manure. So we then started looking for very specific beta-lactam resistance genes, and we looked at CEP02, 05, 04. And what we found was that there was really no difference in the soil. This is quantitative PCR detection of these genes. With 02, 05, no real difference in the treated versus untreated soil. But with CEP04, which is one well-known beta-lactam resistance gene, we found quite a significant increase in resistance in the manure-treated soil. So that suggested that it wasn't just a general resistance, but that it was a specific gene that was somehow increased in these in the soil. And so what we, of course, presumed or expected or predicted was that that was a gene coming from an organism in the treated manure. But what we found was that in the barn manure, so this was before it ever reached the soil, the gene of interest was not actually elevated compared with, so the CEP04 was somewhat elevated in the manure, but not as much as we actually expected in the barn manure. So in fact, it wasn't even statistically significant. So this kind of was a puzzle. We were seeing this increase in the CEP04 gene in the soil when we treated with manure, but we didn't see a substantial increase or significant increase in that gene in the manure itself. So that made us wonder if our original hypothesis that the manure was contributing this gene was in fact, sorry, something funny with my slides here, was actually the cause. So just to summarize, what we actually found was CEP04 showed up in metagenomic libraries from soil, only infrequently from manure, and that was true in the qPCR as well. And the CEP04 increased dramatically after manure treatment in soil. And what we found was it was independent of antibiotic treatment of the cows. So when we used a pristine sample of manure from cows that had never seen cephalothin or any beta-lactam antibiotic, we still saw that increase in the CEP04 gene in the soil after treatment with the manure. So we tracked it down, and what we found very surprising and interesting was that the CEP04 gene is actually derived from a Pseudomonas strain that we could culture from soil. And the Pseudomonas population undergoes an enormous bloom after manure treatment. It's one of the most responsive organisms in the soil to manure treatment. So instead of our prediction that treating with antibiotic-treated manure would increase antibiotic resistance in soil because of either the transfer of antibiotics or genes to the soil, in fact, it was the effect simply of the nutrients brought in by the manure that increased growth of this broad Pseudomonas population that carries a particular resistance gene. So there was clearly an increase in the population that was just accidentally increasing the presence of the CEP04 gene because it was independent of treatment with the antibiotic cephalothin. So this was, we thought, a beautiful example of the use of functional metagenomics to actually find not just resistance, but the resistance gene that was responsible for this bloom in resistant populations. We also found in the Alaska site some fascinating proteins that I'll describe to you now in a search for aminoglycoside and beta-lactam resistance. So we made metagenomic libraries from soil from the isolated remote site in Alaska. We then selected for kanamycin resistance and imipenem, and then began to characterize the genes that we found. I'm sorry, this is both Alaska and West Madison soil. So there are many mechanisms of aminoglycoside resistance. You can get modifications either of amino groups or hydroxyl groups at various sites on the compound, and these can be either acetyl groups or phosphate groups. And we found an abundance of acetyltransferases in our metagenomic libraries that generated resistance through the acetylation of the antibiotics. So this was from the West Madison soil. And what we found was a surprising frequency in the kanamycin resistant strains or clones of these fusion proteins that seem to have two different acetyltransferases fused together. And so this is an example of three different clones that we obtained from West Madison soil. They're definitely different. They're not identical sequences. They're not identical structure. You can see two of them have gaps between the first and the second domain, and one is fused. but they all have the same structure of highly similar aminoglycoside acetyltransferases of two different groups, but within the clones, among the clones, very highly related to each other. So they're not actually that close to those known in the databases, at least at that time, but as you can see, they're above 90% identical to each other, but they vary in the absolute structure as well as sequence. We also found an interesting one that was clearly a fusion. It was actually translated as a single fusion protein. And when we separated the two parts, we found an interesting change in resistance. So when we had the entire bifunctional protein produced by the clone, we saw kanamycin resistance. When we had the left-hand end, which is the aminoglycoside acetyltransferase type, we also saw resistance. But when we isolated the GNAC type of acetyltransferase, we found there was no activity. So it appeared in this case that only the classic aminoglycoside acetyltransferase in CAN4 was required for resistance. And then we found an even more unusual bifunctional protein. This is now from Alaska, and this is resistance to beta-lactamases. In this case, we found an operon structure that appeared to carry a full-length gene that gave us high-level resistance to amoxicillin and piperacillin. Interestingly, the original clone, before we took this gene out, had also resistance to cephalexin, and we still haven't quite characterized that difference, but we think that there's something in the regulatory domains around this functional gene that changed the nature of resistance. But we found that large gene was a fusion of two different types of beta-lactamases, one from class D beta-lactamase and one from class C homologs. And when we isolated the class D, we found that it conferred, very much like the full-length clone, resistance to amoxicillin and piperacillin. But the class C homolog conferred resistance to all three of the other antibiotics, piperacillin, cephalexin, and ceftazidime, which the original clone nor the full-length protein conferred resistance to. So somehow when this domain is by itself, it has a different spectrum of activity. It loses the amoxicillin resistance and it gains the ceftazidime resistance. These similarities are relatively low at the amino acid level. The identity is in the 50% range. So these are rather new proteins, and these bifunctional structures, the fusion of two proteins that form these double functionalities, are quite unusual in nature. Before this study, they've become more common as people have begun to look at more metagenomic clones. At this time, this was the first reported bifunctional beta-lactamase. So the third area that I wanted to talk about is a work still in progress that we find quite interesting and surprising, but it's a challenging story to complete because there's a lot of inference. And as an experimental biologist who's used to doing experiments in controls, it's hard for me to make inferences and evolutionary arguments comfortably. So this story starts in this saltern in Puerto Rico. It's the Candelaria region, and so it's a very high salt saline environment. And what we found there is a beautiful microbial mat. If you're a fan of microbial mats, then you should definitely go to Cabo Rojo and see this mat. Quite stunning. These are evaporative salterns, which is why the salt concentration is so high. Start with salt water, evaporate it, and increase the concentration of the salt. The mats have a laminated structure. I'll show you in a minute. They're about 2.5 centimeters thick, and they have these very distinctive green, pink, and black layers you can see here. So we took metagenomic DNA from the entire mat mixed together. We constructed libraries with relatively small inserts, and then we selected on antibiotic, double antibiotics, both for kanamycin resistance as well as gentamicin resistance. And that turns out to be an important feature of this finding. When we look specifically for genes that conferred resistance to both kanamycin and gentamicin, we found an interesting structure. We found, first of all, a predominance of 16S ribosomal RNA methylases. And so the first one we found was similar to one from a Micromenospora, which it's not very similar, 27% similar, but that was its closest relative in the bacteria. But it's a classic member from all structural analysis to the rRNA methyltransferases. But what came up as really interesting was that when we began to look at the flanking genes on these metagenomic clones, we frequently found that there were not only the methylase, but then flanking genes contained archaeal genes. And so in this one, we see pelota, which is a translation factor from archaea, and also it has homologs in eukaryotes, but it's never been observed in prokaryote, in bacteria. And then we also, on the other side, see transcriptional regulators from archaea, from a nanoarchaeum. So that got us quite interested. We thought, okay, is this a chimera that was constructed in vitro? We did every test we could think of computationally to look for changes in sequence, GC richness, or codon use. We did not see that. There was no evidence of a chimera. There were no obvious restriction sites, similar to the ones we had used to make the clones in the first place. And we found actually more clones that had a linkage between pelota and methyltransferases. So this became a repeatable pattern. So our hypothesis at that point, and this is where the inference comes from, is that the DNA fragment carrying this bacterial methyltransferase is actually derived from an archaea, but has resided in the bacteria for a long time. So it is actually functional there and can confer resistance to antibiotics as well as modify ribosomes. And so then we started looking in other metagenomic clones that we found in Alaska and in the Connecticut manure site. And we found, in fact, some very similar structures, that there were many methyltransferases that specifically, if we selected for both kanamycin and gentamicin, and once again, we don't know why that double selection is so important, we would turn up these methyltransferases, and they all appeared to be from bacteria, and many of them did have flanking genes from bacteria, so that would not be too surprising. But then we started looking for similar homologs in archaea, and we found that, in fact, in the databases, there are quite a few examples. So this is a putative RNA methyltransferase. We were unable to do expression studies in archaea. We couldn't get this gene expressed. So we don't know if it's functional in archaea, but we do know that it resided there and was actually effective when it was cloned into bacteria. It would confer resistance to antibiotics. But interestingly, the flanking genes were classic archaeal genes. And so this is just a really amazing structure, that we have this putative methyltransferase that looks very much like the ones we were finding in bacterial genomes, but entirely flanked by archaeal DNA, and one of them was found in cultured, actually, nanoarchaeum genome. So when we looked more broadly in the databases, there's an example from the Galapagos, another one which is interestingly saltwater, like our original site, the Saltern in Puerto Rico, but then also in a freshwater site in Lake Washington in Seattle. And both of these that we found had 16S methylases, so rRNA methylases, and they were flanked by archaeal functional genes. So this one is a transposase that was found in an archaeal genome. This one is a hypothetical protein from an archaeal genome. And here again, we have the bacterial methyltransferase flanked by a histone acetyltransferase from an archaea. So this raises, but does not answer, several questions of where do these genes come from and where do they go? The marine ones are of particular interest to us because we have repeatedly found this archaeal bacterial linkage. This is one that we really like because there's a transposase from the IS4 family right downstream of the RNA methylase. And so we wonder, again, if this is related to the fact that we find these genes in bacteria, in bacterial genomic DNA, but they appear to be linked to archaeal genes. Here is another one from the marine environment where we found the 16S rRNA methylase from, we don't know where it's from. It certainly has the best similarity to Erosia flexus bacterium. Again, pretty low similarity, but clearly a methyltransferase. And this one is linked to an archaeal protein of unknown function. So we wonder if archaea form a natural reservoir of methyltransferases, and in fact, if these methyltransferases are transferred to human pathogens, could be producing antibiotic resistance in human pathogens that ultimately were derived from archaeal metagenomes. Again, the question for me is, how do we establish this route of movement of genes from the archaea to the bacteria? The genomic structure, however, I think is suspiciously suggestive of a transfer from archaea to bacteria. So in summary, the soil resistome and the water resistome has led to some really interesting discoveries with functional metagenomics. In manure, we found through metagenomics a very interesting antibiotic resistance gene, CEP04, that gave us the surprising result that the manure causes a bloom of a Pseudomonas carrying this gene that's already in the soil pre-manure treatment. And so the abundance of resistance to the cephalothin increases, but that's independent of exposure of the cow to cephalothin, because untreated cow manure will have the same effect. We also found bifunctional resistance genes that apparently have fusions of either aminoglycoside resistance domains or two bifunctional beta-lactamases fused together. And then evidence for the interdomain transfer of methyltransferases that confer resistance to both kanamycin and archaea on E. coli and may have come from archaea originally. I'm sorry, they confer resistance on E. coli. I hope I said that right. So I'd like to thank the people who did this work. Nikolina Udikovich-Kolik did most of the work in the manure system in collaboration with Fabienne Wickman, the two postdocs that were in my lab at Yale. Many other people worked on the West Madison site. The Cabo Rojo site was work done by Gabriel Lozano, and it was originally cited, and the collection's done by Carlos Rios, our collaborator in Puerto Rico. Kristen Riesenfeld started the Antibiotic Resistance Project. Heather Allen and Justin Donato worked on several of the fusion proteins that I talked about today. And Luke Moe also worked on fusion proteins. And with that, I will acknowledge my little friend who was found sleeping in the prairie behind my house this morning. And we're taking suggestions for names for our newly born little fawn. So thanks to all of you for having me, and especially for having me from a distance. And with that, I'll stop and take questions.
Thank you, Joe. And so we have time for questions. And I would say also, those of you who are joining us virtually, if you put your questions in the chat and we'll be able to ask for you. I think we have microphones.
Sure. Hi, Joe. Don Cowan here, University of Pretoria. And by the way, thank you very much for examining that PhD of mine so many years back. So far back that both you and I have forgotten it. A couple of questions about your pristine Alaskan site. I mean, we did a study on some very, very remote Antarctic samples and got a whole bunch of really deeply rooted, what looked like ancestral antibiotic resistance genes. And I just wondered, given the remoteness of this site and the fact that it's had a bit more animal input, but no human input, whether you found something similar in terms of gene diversity, just because of its very low impact in general terms, particularly with human population.
Yeah, I'm actually a little bit skeptical about pristine. I think it's probably today all relative, and maybe it's more pristine than a dairy barn in Connecticut. But with the movement of birds across the globe and the amount of antibiotic resistance genes and antibiotics even that they carry that have been demonstrated to spread across the world, Alaska is on a very powerful migratory bird route. I find it impossible that there isn't some contamination. So I should say that before anything, that of course there is less antibiotic and less human exposure than in an urban site, but I doubt that any of our sites are entirely isolated. We did find some beta-lactamases that looked like they were almost primordial ancestors. That's of course an inference. We can't really prove that. But I have a theory on beta-lactamases generally, that I think they are some sort of primordial enzyme because they tend to be very sloppy in their acceptance of substrate. And I think there's reason to believe that they came probably pretty recently from one of these primordial enzymes that spawned many different kinds of enzymes. So one of the ones we found actually had great similarity to beta lactonases. So enzymes that have similarity, or that have the ability to degrade homoserine lactones. And so I think there's probably a common origin there. That's not a very tight answer, but yes, I think there is evidence that some of these sites have the ancestral proteins. I'm not convinced, though, that the pristine sites are more likely to contain those than the more disturbed sites. And I'd be interested in your thoughts on that, if you think there's any reason that these less disturbed sites would have either a greater diversity of antibiotic resistance genes or the more primordial or ancestral types. I think the soil has tremendous diversity wherever we go. And the disturbed sites seem to have a different diversity, but it's very, very high diversity.
Yeah, thanks, Joe. Yeah, my comments are heavily skewed thinking of the study that we did. I mean, these were truly pristine remote sites, had probably never seen human footprint, and given that they're remote Antarctic sites, no animals, birds, or anything else, nothing overflies, so they would probably one of the last remaining truly pristine types of environments, and therefore I think it's fair to say that what we found there was what had evolved there from however long back you wish, with very little input contamination. Maybe not.
Interesting.
Thank you. Hi, Joe. I'm Gabrielle Potocki-Veronet, and thank you for your great talk. I have a question on this gene transfer that you presented us. To this, by focusing on this super example of gene transfer between archaea and bacteria, and we've often observed such horizontal gene transfers in our own functional metagenomic studies. I was wondering if you have an idea of the frequency of these gene transfers, and does it depend on the gene functions?
Yeah, that's a fascinating question. So you believe me, huh? That's interesting because I'm still skeptical about the gene transfer hypothesis because we can't test it. Yeah, the curious thing is that we have only seen this kind of hybrid structure between bacterial and archaeal genes in the methyltransferases. So we think that there might be something about them that has made them mobile, or maybe there was one event that then ended up with the gene or a homolog spreading throughout the bacterial domain. Or maybe there's something about these genes or the genes that reside near them that make them more likely to transfer. We don't know. I'd be very interested if anyone has ideas about that, but we have never seen that kind of hybrid bacterial archaeal structure with other antibiotic resistance genes.
Okay, thank you.
Other questions?
Hi, Joe. Don here again. This is a question about the example you gave of the bifunctional proteins, the first example where the N-terminal domain was functional and the C-terminal domain wasn't. Why would evolution retain something like that? Obviously, the C-terminal domain presumably has some other functionality which you haven't determined, like stabilization or whatever. Just wondered if you had thoughts on the evolutionary relevance of that. Why hasn't it been deleted if it was non-functional?
Well, I think the first cop-out answer is that I think evolution is still in progress. So the question is, maybe this gene in other settings, that domain has been lost because it's not functional. It also, as we saw in the later example, may change the spectrum of resistance. So maybe there is a selection for it, and we just didn't test the right antibiotics to find that difference in spectrum of activity. Or it may have an entirely different function. Like you said, it may confer stability. We also found a very peculiar fusion of a sigma factor with an antibiotic resistance gene. What's up with that, right? What's the value of fusing a coding protein that makes one end a sigma factor and the other end a functional enzyme? I haven't seen other examples of that in the literature, but it is a true fusion. We actually showed that the protein that's produced, in fact, Justin Donato, who I believe is at the meeting, showed that the full-length protein is partly the sigma factor and partly the resistance gene. So I don't think we know a whole lot about these bifunctional proteins because they aren't found in the frequency that we find them in metagenomic DNA from the environment in cultured organisms. And so much, of course, of what we know about protein structure is from the cultured organisms. There are very few, if any, I think, in E. coli of these bifunctionalities. And so I don't think we know how these evolved or where they're selected for. I mean, one problem we always have is that we sample at one point in time in one environment, but that same bacterium may have experienced many other selection pressures that aren't evident from where and when we sampled.
Cool, thanks.
Us?
Hello, thank you, Professor Handelsman, for the talk. And this is not my area of expertise, but circling back to the transfer of methyltransferase potentially between bacteria and archaea just made me think of another PhD student's work in the group who works with metaviromes and viruses. And I was thinking if there is any evidence that, common viruses and those, I literally Googled, I found a paper that says there are those viruses in hypersaline, like Great Lakes, that interact with both archaea and bacteria. And yeah, so potentially, do you think that might be causing the lateral gene transfer?
Yeah, that's a great hypothesis. And I think I saw the paper that you're talking about. And I think viral transfer is probably our best bet for explanations. We found, as I mentioned, IS sequences, but not evidence of phage or viruses nearby. So I wish I could tell you we had data for integrases that look like phage functions, but so far we haven't seen those.
So I'd like to follow up on that point a little bit. Just in general, do you have any thoughts on for transfer that we see evidence of in genomes, what the major functions are, or the major mechanisms are?
Mechanisms of the
mechanism of transfer.
Oh, mechanism of transfer between bacteria and archaea. I don't know. As the last questioner commented, there is evidence that there are viruses that infect both bacteria and archaea, but it certainly could be a transformation process or something, or who knows, perhaps there's even conjugation across domains. We've certainly seen conjugation between bacteria and eukaryotes, so why not between bacteria and archaea? But I don't know of any evidence of that. If anyone else has seen evidence of that, I'd love to hear it.
Yeah, I think the evidence is the challenge.
Yeah.
Do we have any more questions? Okay. Put this question. Okay, great.
Does it? Okay, now it works. So my name is Alexander Wenzel from Sintef in Norway. I have a question about the general diversity of resistance genes in nature. And many of the antibiotics or the bioactive compounds that we find are in clusters, which also include resistance genes against a particular compound in some extent. Can you comment on the diversity and the potential for finding new antibiotics that then immediately lead to a resistance because the resistance gene is still around?
That's a question we're working on right now, really, really hard using computational approaches. What we found so far is that there's a surprisingly low, and I don't mean really low, but lower than expected frequency of the linkage of resistance genes with biosynthetic pathways. When we started this work, we were very hopeful that the resistance genes would lead us to antibiotic biosynthetic pathways, partly because that's how we found the zwittermycin biosynthetic pathway. We cloned the resistance gene, and sure enough, downstream of it was, and upstream was a 65 kb region responsible for biosynthesis of zwittermycin, and that opened up the entire biosynthetic mechanism. And so we went in naively thinking, well, we'll find that again if we look for broadly active antibiotic resistance genes. But our recent computational analysis suggests that, in fact, many pathways do not carry the resistance gene linked to them, as we would have predicted for most antibiotics. And many resistance genes, and we don't have exact numbers, so I won't cite it yet, but somewhere I would say around a third to a half of the resistance genes are not found linked to biosynthetic pathways. So we should have found more biosynthetic pathways than we did, because we didn't find any linked to our resistance genes. based on those data. But of course, those data are based on the database metagenomes and not necessarily all the ones that are out there that we're sampling. Whether our samples of metagenomes in the databases are representative of what's in nature and of any given sample that any of us takes from the soil or from manure or any place else, I don't think we know. So that's something we're pursuing. I can't give you a better answer than that, but we were surprised at the lack of linkage, the lower frequency of linkage, both in the metagenomic experiments where we didn't find any biosynthetic pathways, as well as in the computational analysis.
Thanks a lot.
Can I just follow up on that? I mean, the obvious anomaly there, an organism generating, or at least with an antibiotic biosynthetic pathway, presumably is susceptible to the antibiotics producing if it doesn't have the resistance gene. The implication then that either it has some other inherent resilience mechanism or the pathway is non-functional for whatever reason. And I know many of them are cryptic and not necessarily expressed. Your comments?
So there are several possibilities. One is that that particular antibiotic does not affect the organism producing it. It just doesn't have the right target. So perhaps it's a non-cell wall containing bacterium that produces a cell wall synthesis inhibitor. Obviously, it's not going to be inhibited by its antibiotic. Another is that I don't believe antibiotics evolved as warfare molecules in many systems in biology. There are, in fact, only a handful in which there's demonstration that evolutionary data show that, in fact, it is killing other organisms or inhibiting their growth. That is the role of those antibiotics. For example, in the ant symbiosis system, I think there's very good evidence that the Streptomyces antibiotics keep the ants' fungal gardens clean and healthy. And so there are, most of our examples of antibiotics are simply compounds that we have studied in the lab at high concentration that inhibit other organisms, but we have no idea what they do in bacteria. My lab is looking right now at the role of antibiotics as signals. This was an idea started by Julian Davies more than 2 decades ago, and we think it's time to really explore that. And we find that, in fact, transcriptional regulation by antibiotics, and this is exactly what Davies found in 2001, transcriptional regulation by antibiotics is tremendous and fairly global across genomes. We just published a paper called Thor's Hammer in MBio a couple weeks ago that showed that if we look at RNA-seq patterns across a model community, we find that one compound, coriansin, produced by one of the members, which is an antibiotic, has global impacts on gene expression. I mean, thousands of genes change their expression direction, go either up when they were down or down when they were up. And this is not just an SOS type stress response. So I think that antibiotics play much more subtle and complex roles in bacteria and are produced, therefore, probably at lower levels than is needed to kill either the producing organism or a target organism.
Yeah, thanks, Joe. I know the chat line, Lonnie Van Sale from UWC in Cape Town has made us sort of comments on that.
Yep, I totally agree with Lonnie.
Hi, Lonnie.
So just so everybody everybody in the room might not be able to see what Lonnie has written there, but he says, could that be an indication that secondary metabolites are not predominantly used as an antimicrobial by bacteria?
Then he says, hi, Don.
But I think there are also other genes in genomes that may confer resistance, and it doesn't make obvious sense why that resistance gene might not be linked to the biosynthetic pathway, because it seems like if they could be separated, there's an evolutionary disadvantage there for sure. but perhaps there's some other selection pressure that maintains the resistance gene. So I think several possibilities explain why there's less linkage than we expected.
Okay, so if there's no further questions, I would like to thank Joe again for an outstanding start to our conference and getting us out on the right foot. And also thank you, Joe, for that extended discussion. I think it really sets a good tone for us. I hope that you're able to join us for the rest of the day. And also, I will thank you for the effort that you made in trying to get here in person. I know that travel is challenging in these times, and it is very much appreciated. And I'm thankful that this virtual session worked out so well. So thank you again.
Appreciate the invitation and your flexibility to have me here virtually.
So we have a break, so we can, there's coffee out there, and there's refreshments, and we will be back, what time? 10:45? 10:45. So we have quite some time to mingle.